Hello, today we are going to discuss the steps of synaptic transmission in a chemical synapse. So, this is the presynaptic neuron, this is the synaptic cleft, and this is the postsynaptic neuron. Okay, so the first step is the arrival of action potential on the presynaptic neuron. Second step is the opening of voltage-gated calcium channels on the presynaptic membrane, especially at the active zone. Okay, so the third step is the entry of calcium via this voltage-gated calcium channels, and it causes the fusion of the synaptic vesicle with the presynaptic membrane with the help of synaptobrevin and syntaxin. So it's the fourth step. It's the calcium mediated exocytosis of the vesicles and the release of the neurotransmitter from these vesicles. So what happens to this neurotransmitter? The neurotransmitter, they binds with the receptors which are present on this postsynaptic membrane. See, if the neurotransmitter which was released is an excitatory neurotransmitter, then it leads to opening of voltage-gated sodium channels and sodium influx, depolarization of the postsynaptic membrane, and development of EPSP or excitatory postsynaptic potential. And if the EPSP is more than the threshold of excitation, what happens? There is a development of an action potential at the initial segment of an axon, and this propagates along the axon to the axon terminal. But if the neurotransmitter release was an inhibitory neurotransmitter, then it causes the opening of chloride channels, and there is hyperpolarization of the postsynaptic membrane and development of IPSP or inhibitory postsynaptic potential. So the action potential cannot be developed over here. So finally, what is the fate of the neurotransmitter in the synaptic cleft? The other neurotransmitter which was left behind, they can either be degraded or diffusion or reuptake. I hope the idea of the steps are clear. Thank you.